In this module, we will be talking about adamantane and its structure, the nomenclature of adamantane, synthesis of adamantane, synthesis of its derivatives, their properties, and their applications in pharmacology and biochemistry. The word adamantane is derived from a Greek word admantinos, which means relating to steel or diamond. Adamantane is a colorless crystalline compound with a camphor-like odor. Its melting point is 270 degrees Celsius. Its molecular formula is C10H16. It contains 3 cyclohexane rings as shown in the figure, which are connected and arranged in an armchair configuration. The molecular parameters were deduced by electron diffraction and X-ray crystallography. The carbon-carbon bond length in adamantane is 1.54 angstrom, and it's almost identical to that of a diamond structure. And the carbon-hydrogen distance is 1.112 angstrom. Adamantane is the most stable isomers among all the isomers with the chemical formula C10H16, which include similar structures of twistane. We will be studying about twistane later. The arrangement of carbon in adamantane is very much similar to that in the diamond crystal. That means all the carbons are tetrahedral in their geometry, and that is why it gives the stability to this structure. The discovery of adamantane in petroleum in 1933 launched a new field of chemistry dedicated to studying the synthesis and properties of these polyhedral organic molecules. Adamantane derivatives have found practical applications as in drugs, polymeric materials, and thermally stable lubricants. In a other wide variety of applications also, adamantane has found its utilizations. Now let us see how we actually do the nomenclature of these molecules. According to the systematic nomenclature, adamantane should be called tricyclo-3,3,1,1 decane. But the IUPAC recommends using the name adamantane for convenience. This molecule is composed of only carbon and hydrogen atoms, and therefore its 16 hydrogen and 10 carbon atoms can be described by only 2 sites as labeled in the figure. The site 1 is equivalent to 4 equivalent sites. As you can say, the site 1 can be seen as the bridged carbons, whereas the site 2 shows the non-bridged carbons and is equivalent to 6 sites. It has on the whole tetrahedral symmetry. Noradamantane and homoadamantane is the closest structure analogues of adamantane, which respectively contain 1 less and 1 more methyl link than the adamantane molecule. Now, how these molecules are synthesized? Adamantane was first synthesized by Vladimir Prelog in 1941 from Meerwein's ester. This process was impractical as it contained 5 stages and had a yield of only about 0.16%, wherein initially the first step involves the cyclization and then followed by reduction of the ketonic groups, followed by final carboxylation reaction, giving you the final adamantane molecule. Prelog's method was modified by Paul von Roeg Schleyer in 1957. Dicyclopentadiene was first hydrogenated in the presence of a catalyst, for example platinum dioxide, and then it is transformed into adamantane using a Lewis acid such as aluminum trichloride at 150 degrees to 180 degrees. This method increased the yield to about 30 to 40% and provided an affordable source of adamantane. Now let us see the synthesis of adamantane's derivatives. There are a variety of derivatives of adamantanes. We will start with protoadamantanes. This can be synthesized by rearrangement of adamantane nucleus. For example, exo-4-protoadmantyl acetate can be prepared after the acid catalyzed deamination of 2-admantyl phenyl triazine as shown in the scheme. The acetolysis of 2-admantyl tosylate give rise to 5% yield of the same acetate. The major product of the unrearranged 2-admantyl acetate was found in both the cases. 4-protoadmantyl acetate may be converted via alcohol to 4-protoadmantone, which gives protoadmantane upon Wolff-Kishner's reduction, as shown in the scheme. While deamination of 2-aminoadmantane 1-ol catalyzed by the nitrous acid provide 92% yield of 4-protoadmantone. Furthermore, 56% yield of 8-hydroxy-protoadmantone was obtained after the deamination of 2-aminoadmantane 1,3-diol as shown in the scheme. Similarly, another way of preparing protoadmantone is the hydrolysis of 1-methyl 2-admantyl tosylates as shown in the scheme, which gives 4-methyl-exo 4-protoadmantol in approximately 30% yield. After the reduction of protoadmantone with lithium aluminum hydride, it gives the mixture of 2:1 mixture of endo-protoadmantol and its exo 4 isomer. This stereochemical assignment is consistent with the relative solvolytic reactivities of these derivatives in alcohols. The addition of methyl Grignard reagent to 4-protoadmantone provide 2:1 mixture of 4-methyl exo 4-protoadmantol and its corresponding endo-epimer. Now let us see the preparation of another derivative of admantanes that are called twistanes. As the name suggests, they have got some twisted bonds in their structure. Like admantane, they also have 3 cyclohexyl structures, but these cyclohexyl structures are arranged in a different configuration as compared to the admantane structure itself. These twistane derivatives can be easily prepared in one step from cis-decalin 2,7-dione as shown in the given scheme in presence of acetic acid, acetic anhydride and a Lewis acid such as BF3 iterate. The wide variety of substituted twistanes can be synthesized by conventional techniques after selective removal of these acetoxy twistanone, that is the ketonic group as well as the acetoxy group. We can convert these twistane into different other derivatives, other substituted derivatives. The multi-step synthetic route was also developed for the synthesis of optically active twistane. The synthetic method also provides route for the synthesis of tricyclodecane as shown in the scheme, wherein a Diels-Alder reaction of methyl cyclohexadienyl carboxylate, which acts as a diene, with methyl propionate, which acts as a dienophile, and it gives a mixture of a bicycloderivative as shown in the scheme. These bicycloderivatives on reduction provides, which on further cyclization provides a twistane molecule. Tricyclo 5,2,1,0,4 decanes. These tricyclodecane can be synthesized by pyrolysis of the sodium salt of tosyl hydrazone of 10-bicyclo 5,2,1-decanone. They have a crown-like structure wherein the 3 rings are fused together. These are called tricyclodecanes. Furthermore, a transannular ring closure can also be used for synthesis of such molecules, wherein as shown in the scheme, the tricycle decanol can be synthesized by a ring closure of a ketotosylate, which is shown in scheme in presence of lithium and ammonia. Now homoadmantanes, they can be prepared by ring expansion of admantanes. Now what are homoadmantanes? Herein again we have 3 condensed ring. It's a tricyclo molecule, but not having exactly the cyclohexane moieties. It may have cycloheptane moieties or some other cyclo rings in their structure, and they collectively are known as homoadmantanes. Now homoadmantane derivatives are most readily obtained from ring expansion of appropriately substituted admantanes. For example, the carbonium ion reactions of admantyl carbonyl systems have been well known route to give 3 homoadmantyl derivatives. The mixture of homoadmantyl acetate, and 1-admantyl carbonyl acetate were obtained after the solvolysis of 1-admantyl carbonyl tosylate in acetic acid and sodium acetate. In more nucleophilic media, for example, water diglyam and NaOH, only 3 homoadmantol is observed. Now 4-homoadmantone. 4-homoadmantone means which contains a ketonic group at position 4, which is obtained from a ring expansion of diazomethane, which is known to have insertion of one carbon atom. The diazomethane molecule is very useful for the insertion of one carbon atom into any kind of carbon-carbon bond. So here this is used for the ring expansion of admantone, and this results in the formation of 4-homoadmantone molecule. Homoadmantane and a variety of 4-mono and 4,5 disubstituted homoadmantanes can also be synthesized by similar ring expansion reactions. Now, the preparation of these homoadmantanes by ring closure reaction. The ring closure reaction is also synthetic route for homoadmantane derivatives. The first involves the transannular cyclopropyl participation in the solvolysis. After acetolysis, only one product, that is homoadmantyl acetate, is observed. As shown in the figure, this is a single step synthesis of homoadmantyl acetate derivative. Now moving on to the other derivatives, we have noradmantanes. This ring closure again is useful for the synthesis of noradmantanes. It involves a transannular ring closure of bicyclo 3,3,1 nonyl system. As we all know that 3,3,1 actually indicates the number of carbon atoms in each of the rings. So you can see the reaction of this 7-methylene bicyclo 3,3,1 nonane with sodium in moist ether provides a noradmantane derivative. Furthermore, a single Ferotsky ring contraction of 1,5-dibromoadmantane 2,6-dione also enables the preparation of 3 substituted noradmantanes. As shown, the first step involves the ring contraction in presence of KOH, where a 6-membered cyclohexane ring is contracted to a 5-membered ring, which is followed by simply the reduction, followed by its decarboxylation will provide noradmantanes. Now move on to another derivative of admantanes, that is bis-noradmantanes. Bis-noradmantane derivatives can be synthesized by Ferotsky ring contraction of 1-dibromo-pa-admantane 2,6-dione as shown, which involves again a kind of Ferotsky ring contraction reaction followed by the reduction and its further multi-step synthesis to bis-noradmantanes. The difference between noradmantanes and bis-noradmantanes is clearly visible as you can see the molecules. In noradmantanes, the 2 cyclopentyl rings are joined by a bridge carbon atom, whereas in bis-noradmantanes, the 2 cyclopentyl rings are directly connected to each other. Dihydroadmantanes and dihydrohomoadmantanes. Here the carbene insertion or 1,3 elimination reaction are widely used for the synthesis of dihydroadmantanes. For example, pyrolysis of the dry sodium salt of tosylhydrazone of admantone gives good yields of 2,4-dihydroadmantane, and by the treatment of this 1,3-dibromoadmantane shown in this figure with sodium and 1,3-dihydroadmantane was obtained. Yet another derivative of admantane is ethanoadmantanes. Herein, a ring closure reaction of a substituted admantane can be obtained by the synthesis of ethanoadmantane, Lewis acid catalyzed rearrangement of various polycyclic hydrocarbons. For example, the carbene insertion reaction illustrated here in this figure during the treatment of 2-admantyl diazoketane ketone with copper. Properties of admantane and its derivatives. Now moving on to the physical as well as chemical properties of these admantanes, let us see. First of all, we will move on to thermodynamic properties. The deriving force for the Lewis or the strong acid catalyzed rearrangement of polycyclic hydrocarbons to adamantane systems is provided by high thermodynamic stability of the admantyl ring system. As we know, because they have a structure which is similar to a diamond ring, their structure is quite thermodynamically stable. So we can say that adamantane is the most stable tricyclic hydrocarbon, and due to this high degree of branching in the ring system and from nearly ideal conformations of the atoms in the molecule, this thermodynamic stability is conferred upon this molecule. Now moving on to the chemical property, the ionic reaction. Although the bridgehead positions of many polycyclic hydrocarbons have been found to be quite unreactive toward nucleophilic substitutions, the 1-admantyl cation can be generated with relative ease, since tertiary 1-admantyl cation is more stable compared to the secondary cation. Means at the bridgehead position it will be forming the tertiary cation, and at the non-bridgehead position it will be forming a secondary cation. So at the bridgehead position, the carbonium substitution reaction strongly is preferred. In the absence of free radical catalyst, the bromination reaction provides high yield of 1-bromo-adamantane as shown in the scheme. In another reaction which involved the initial generation of 1-admantyl cation, either by means of hydride transfer to the tertiary butyl cation or from 1-bromo or 1-hydroxy-adamantane, followed by trapping of the cation by a carbon monoxide and acetonitrile respectively. This may be used to introduce a carboxylic acid and amino functions in this admantyl moieties. Now 1,1-dichloroethylene and 1,1,3-trichloroethylene can also be used for the trapping of 1-admantyl cation. We can use these moieties to trap this cation to give 1-admantyl acetic acid in the former case and alpha-chloro 1-admantyl acetic acid in the later case respectively as shown in this scheme. Now free radical reactions of admantanes. In contrast to the specific ionic reactions of diamonoid hydrocarbons which are discussed earlier, the free radical substitutions are much less selective. Thus, the free radical reactions provide a method for the preparation of a greater number of possible isomers of a given hydrocarbon that might be available by ionic process. It is because of the stability of the specific cation as compared to the free radicals. Generally, it is difficult to separate the complex product mixture in case of free radical reactions. Consequently, there are few examples of the synthesis of specific derivatives of diamonoid hydrocarbons by this method. For example, photochlorination of adamantane in CCl4 gives 37% of 1-chloroadamantane and 73% of secondary isomer. So due to the decreased symmetry of the substituted admantanes, free radical substitutions of these substrates give rise to even more complex product mixtures. Occasionally, special circumstances permit a simple separation of the products which result from such free radical substitutions. Thus, chlorocarbonylation of adamantane followed by methanolic workup as shown in the following scheme results in a nearly 1:1 ratio of 1 and 2 methyl adamantane carboxylates, which may be separated readily by fractional distillation. So how can we prepare 1,2, 1,4, 2,4 and 2,6 disubstituted admantanes selectively? Let us see. While the preparation of bridgehead, mono, di, etc. and methyl substituted admantanes is extremely easy, polysubstituted admantanes in which one of the substituents is at the methylene position are much less readily prepared. In general, special techniques must be developed for each specific substitution pattern. For example, 1,4 disubstituted derivatives can be easily prepared from the 2 substituted admantanes. That means we can begin with the 2 substituted admantanes and then we can convert it into 1,4 disubstituted derivatives. It provides a convenient precursors for the preparation of 1,4 disubstituted derivatives. For example, the deactivation of the adjacent bridgehead positions by methylene substituent directs the ionic substitution to other bridgehead sites. Thus, for example, in the given scheme, the treatment of adamantone with bromine in presence of aluminium tribromide for 10 days gives 5-bromo 2-admantone in 90% yield, which is quite a good yield. The internal cyclization reaction of a bridgehead substitution provide 1,2 substituted admantanes. 1-hydroxy-2-admantane was synthesized by the photolysis of azidoformate as shown in the scheme which is in front of you, followed by internal cyclization and the formation of nitrene intermediate. Furthermore, same type of internal cyclization occurred during the reaction of 2,1-admantyl ethanol and lead tetraacetate or with HOCl to give 1,2-disubstituted admantone. The 2,4-disubstituted admantanes can be prepared by the ring closure reaction of bicyclic nonanes. Thus, as shown here, the lactone can rearrange in 50% sulfuric acid to give 4-hydroxy-2-admantane via the acylium ion. Similarly, the skimmed reaction of admantone results in an overall formation of 4-methyl sulfonyl 2-admantone, presumably via the unsaturated nitrile. As shown in the scheme, you can see we are reacting admantone derivative with sodium azide in presence of methyl sulfonic acid, which is giving a nitrile as an intermediate, and then it rearranges to give you a 2,4-disubstituted admantyl. Now moving on to 2,6-disubstituted admantanes. 2,6-disubstituted admantanes may be obtained by the direct synthesis or as a minor component of specific ionic substitution reactions. The treatment of 2,6-bicyclononadione with pyrrolidine and methylene iodide followed by acid hydrolysis provides pure 2,6-admantane dione. This is a very good simple one-step synthesis for 2,6-admantane derivatives. Now moving on to its practical applications in pharmacology and biochemistry, these admantane and admantane derivatives have been proved very versatile. The applications of admantane derivatives in the field of pharmacology have given added impetus to research in admantane chemistry. Numerous derivatives have been shown to have antiviral activity, whereas the efficacy of other known drugs has been found to be significantly enhanced or modified in a useful way by incorporation of admantane moieties. The 1-admantane amine hydrochloride salt is useful for inhibiting the specific stains or influenza such as A, A1, A2 and C in tissue cultures or chick embryos or in mice, and the range of effectiveness of this drug has been considerably extended and similar antiviral activity has been found in humans as well. The structure-activity relationship also demonstrated that by an alternative experiment, we can make these derivatives to be useful in other biological activities. For example, the sedative property of a drug requires the drug to be transported directly to the nervous system. Therefore, a decrease in the hydrophobic bonding should result in an increase in its sedative activity. The successive introduction of methyl groups to the admantane carboxamate moiety, it increases the sedative activity of the drug as shown in mice. The admantane substituted naphthoquinones given in the figure is found to have effective anti-malarial activity. Similarly, other admantyl esters of pyridoxal hydrochloride nature, little complex molecules, but you can see it in front of you, they are found to have a growth inhibition property. In another case, the 1-admantyl chloroformate and 1-admantyl chloride has been shown very useful blocking agents in biochemical synthesis. So, there are more applications to be considered and explored for these kind of moieties, and the research is still on to find the other applications of these admantyl moieties and their derivatives. Now, let us summarize what we have studied in this module. The chemistry of adamantane and related cage-like hydrocarbons has developed rapidly in recent years. That is what we have seen. These cage-like molecules are very important in having practical applications in, you can say, pharmacology, where the drug has to be delivered in a specific, enclosed in a specific cage-like molecules. This provides a good platform. These molecules provides a very good platform for such applications. The chemistry of such systems is typically hydrocarbon in nature. They are purely hydrocarbon in nature. Yes, we can substitute it for getting the required applications as and when are required, and we can explore it further. And the particular utility of these cage-like substrates in a variety of chemical investigations arises the need to study these molecules in more detail and exploit their properties in a wide applications. The adamantane and their derivatives are widely used in various biological activity, as we have discussed in this chapter.